I'd like to thank you again for joining us and thank Rob for making the time to put this together. Everyone is on mute and will remain on mute. You can type in questions for Rob to the chat, to either Rob directly or preferably to Macmillan Education or Scott Guile, and we'll read them at the end of the presentation. Rob Liu is a team teacher of Life Sciences 1A at Harvard. It's their introductory biology and chemistry course. It's also one of their largest courses on campus. He's also the director of the Derek Fox Center for Teaching and Learning at Harvard. He has put together award-winning animations, including The Inner Life of the Cell and many of the animations that go along with the Morris et al. Text from Macmillan. And with that, I'm going to turn it over to Rob. Thank you very much.
Okay, thank you, Scott. Welcome, everyone. I'm really honored that you're taking this time to join me. So what I thought I would do today is sort of share with you some of my adventures, if you will, with using technology, particularly as it relates to using visual materials as sort of a driver for student learning and in some ways student motivation as well, but to also help us think about how do we actually use what we learn from a variety of technological platforms to really understand how students are learning and to really understand how we're able to, in fact, enhance the student experience much more effectively. Can folks see my desktop yet?
Yes, we can see, if it's your biography that's up, then yes, we can.
Okay, but can you see Launchpad right now?
No, you need to, you still need to share your desktop, Rob.
Okay. Sorry about that.
Not a problem.
Okay, there we go. So what I thought I'd do is to spend a little bit of time talking about how I've been able to use, for example, various kinds of visualizations to really get students engaged, and in particular, how that really allows me to diagnose what's happening in the classroom and how well students are able to synthesize concepts. So what I think you should be seeing right now, I believe you can see my desktop. Am I right, Scott? Is that true?
Yes, we're good. The PowerPoint was kind of small, but what you're showing now is full screen.
Okay, fantastic. So what I'm trying to do right now is sort of give you a sense of how using some of the resources that my colleagues and I, my co-authors and I, in How Life Works, how we've used that as a way of engaging students, but also driving some of the assessment structures that we have. So what you're looking right now on Launchpad is essentially what we have in terms of not only the ebook, but a series of resources for the book as well. But what's really important is the way in which visualization can be used as sort of a way of assessing what students know and what they understand. So just to jump in sort of a little bit, one of the critical features of what we're going to do is to take a look at, and it's something I do with my students all the time, is to have them use visualizations as sort of an important starting point. So what I've just done is to open sort of a resources list, and one of the key resources that we have is really looking at what we call visual synthesis maps and how those maps can actually allow us to, in fact, explore various aspects of biology in a way that in some ways is more student-driven and more motivating. So that one of the maps that we might look at, for example, is one that unpacks gene expression. So I'm clicking on that right now. And so what you can see here is that what we have is something that almost looks like a Google map version, if you will, of sort of one way of exploring the cell. And let me switch to full screen mode so that you may be able to see this a little bit more clearly, right? And so what you have here is a map that connects exactly with a textbook diagram, what we call a visual synthesis in the book How Life Works. And one of the things that we're interested in doing is how can you take a diagram and not only bring it to life, but how can you allow students to explore it in some really interesting and perhaps novel ways. So one thing we're trying to do is to take advantage of students' interest in sort of compelling science visualization and use that as a starting point, if you will, for how they can explore what they're looking at. So what you're looking at right now is in fact a map, but what is also important is that we have actual animations that go along with it. And so what I'll do is I will bring this down sort of a little bit so that you don't need to hear me talking twice. But with this animation, what this allows us to do is to use the process of visual representation as a way to unpack what's happening ultimately in a biological process, but to give students more agency in terms of understanding what's going on. So what I'll do, in fact, is just play briefly, without the sound, what we're going to hear, what we'd actually see, if you will, taking a look at this animation. So that, of course, as we go through a variety of processes, as we have showed sort of very briefly, examples of translation, for example, and transcription, what this allows us to do is to dive into the cell and in a way give students an opportunity to not just look at things and think, oh, wow, how beautiful is that, but it gives us an opportunity to interrogate their sort of ability to tie things together. So for example, one can look at this sort of view that we have right here, and we can really ask them questions, for example, of what are they looking at based on what they understand of the cell, what they understand of the various components, how can they translate what is really a representation of what they might find inside the cell? What are these various components? How do we start to unpack the process of representation? How do we, for example, look at this image and say, well, what are you looking at right there? Not just at the gross level, but if they may recognize something that's the endoplasmic reticulum, what do we think are those little dots that they see on the endoplasmic reticulum? What is this structure that they see moving around in the cell? Why would that structure actually move? Once you go into the nucleus, what are they looking at here? What is all of this tangled material? So that what we're trying to do, and what something I practice a lot in my class, is to in fact use this as an opportunity to not just simply tell them things, but to show them things and have them interrogate what an actual representation might mean. So I will show this in lecture, and then, for example, looking at what we're seeing right now, I will ask them, what do you notice, about this representation? And of course, those that are paying attention will realize that there seems to be a more densely packed region. And what is that densely packed region, right? What are all these structures that we see moving around? And what is it about that representation that we think is not only accurate, but what are some of the tricks that have been done visually to make this understandable as a representation? So that one of the things I'm very interested in is how do we use the technology of visualization to not only show students things, but to allow them to take a more active role, in fact, in unpacking what that representation actually means. So that little sort of animation preamble can lead us into sort of exploring what you're now seeing on screen, which is an actual visual synthesis map focused on gene expression. So that students are accustomed to seeing this kind of diagram, and what you'll see is that it actually connects with the animation that they just saw. Here is the endoplasmic reticulum. Here is that density, of course, in the nucleus, which hopefully they will connect with the nucleolus. And this allows us now to either individually for individual students, but in particular in the lecture, to actually start to go through this. So when we first designed these visual synthesis maps, I originally conceived of them as being something that students would use back at their rooms. That maybe it would be a study tool that 1 or 2 or 3 students would cluster around, and it would give them the opportunity to explore. The very first time we showed this to a group of fellow instructors, I still remember that moment when one of them said, "But Rob, what you've done is for me create what will be the ultimate replacement for PowerPoint," right? That this will be sort of an absolutely fantastic way for us to use sort of a visual support that is not just a slide, but will actually allow you to explore in a more sort of directed way with students, and a more flexible way with students, a process like gene expression. So I will load this in the classroom, in the lecture hall, and I will actually project this. And what we'll do is we'll have a much more active exercise where we'll actually begin with a simple discussion of what these various components are. As you can see, there are no labels. So students now need to figure out, what are these things that we're looking at? We know it's a cell, but what could these things be? And what I will typically do at that point is have them lead, for example, the path of exploration that we might take. So that looking at this sort of specific sort of image, we might say, okay, what are we interested in exploring? So they might say, well, what is this dense structure right there? What might that be, and what does that look like as we zoom? So that you can actually zoom into it. And as you zoom into it live with the students, and as you're seeing now, it allows you to get ever closer, and as you can see, you start to see more and more detail. So this allows us to place in the context of the cell what these various components are, what this dense tangle of chromatin actually is, and then start to have a discussion of how are these things so very densely packed, so that they can now bring together and zoom in and see that, well, what we're talking about DNA, it really gives you the opportunity to reinforce the issues of base pairing in the context of the double helix, but at the same time, it also allows you to now discuss with your class in real time the roles of proteins in this process of packing ultimately, right? How do these histone sort of cores actually allow DNA to bend? How is it, for example, that this actual structure allows what one might expect to be an energetically highly unfavorable process to take place? And that really allows you to sort of tap into that idea very much on the fly with them, but at the same time in a manner that allows you to then zoom back out and put everything that you've talked about in context. So that it's a way for you to work with your class in a way that gives them further agency. And that at each, for example, zoom step, what you will then see, of course, as one might notice here, is that, well, lo and behold, there are particular genes from this dense packed set of chromatin, this dense packed nucleolus, and what do those encode? They don't encode proteins. What they in fact encode are transfer RNAs, that particular gene. And that allows you to then discuss transfer RNAs, what is the role of transfer RNAs, the fact that they are critical in terms of the assemblage of amino acids in translation, that those amino acids must be loaded on individual transfer RNAs, and that those transfer RNAs in turn play a critical role in translation itself. So once again, now this allows you to connect it in the same context of the cell with the literal process of translation. You can then say, okay, let's actually review codons, right? Codons and anticodons. It allows you to go back to sort of the actual phenomenon of base pairing. And so once again, it allows you to tie together multiple concepts in some very interesting ways. So this is one case in which one might argue that the simple technology of Google Map and that idea of a Google Map allows us to explore something that is really compelling and that gives students, in the context of a class with you, a way to guide themselves and to choose what they might want to explore. And so, for example, here as well, Then there was that tRNA gene that we looked at that's being transcribed to give you tRNA. But then what's happening over here? Here we have another transcription process that's taking place. But now, of course, this is a protein coding gene, and this allows you to get into the issue of RNA splicing and lead them through it. So that once again, it gives you enormous flexibility in the classroom without having to, if you will, be fixed by what the particular sequence of PowerPoint slides might be. So that's all good and well, but what is really advantageous here as well is that you can do this with your students in the classroom, but of course, this is also available to them in the privacy of their own rooms. So you can actually assign something like this in the context of Launchpad so they can then go into this structure and explore it either on their own or with fellow classmates in any sequence that they want. So for example, you might have taken the advantage of this opportunity to explore tRNAs, where they come from, and connect them back with the process of translation, but you may not have touched, for example, on splicing. So now they can go back and you can actually assign them with the task of going back. You can actually assign questions associated with the visual map and have them, in fact, explore splicing on their own, not simply through questions, but in a way through the visual map itself. You might actually look at this and think, well, what will happen if they all get lost? One advantage of this sort of approach is that there's always a location bar, a location box right here, so that in the lower right, so that no matter where you go, as you can see, the location box tells you where you are, and you can in fact use that little guide to also move yourself around. So the location box is always there. You can always reset, but then there is a navigation menu that you can turn on. And so let's say a student is interested in going back and thinking about translation or thinking about RNA processing. When you click on any of those, it gives you the opportunity to then dive into that very specific topic. So that what we're looking at here is the full screen mode, but this is what students would see in their room. And as you can see here, we have that location sort of map. We can turn that off. Here we have the navigation map. So you click on transcription, and what this allows you to do is to see key transitions. So you click on that transition. Each one takes you to a specific, particularly important transition. This one that allows us to connect RNA polymerase with the process of transcription specifically, showing the transcription bubble, tying it back to base pairing, and one can look even more closely on the incoming nucleotide that's being added, right? So that this allows you to, in fact, really explore this. You can also go back and look, for example, at a more complex view of how, for example, packing actually takes place. So this allows you to connect simulations, various high points, and ultimately, though, they can go back to the overall map, turn all of that off, and explore freely on their own. But what you really want to do is to be able to tie this to assessments, right? So you can begin a visual synthesis map assessment. And what you'll see is that you can create and assign your own series of questions. And here's one example where, a very simple one, where does the RNA polymerase initially bind? Binds at the promoter. We click submit. Their response is saved. And you can then go on to the next question. Where would you expect to find unprocessed RNAs? Let's say if you said by mistake in the cytoplasm, you submit, you have the opportunity to try again. You say in the nucleus, and once again that response is recorded, but you can zoom to the place in the map that actually deals with that very specific topic, go further in, and this will allow you, for example, to really explore those ideas more effectively, and to also work perhaps with other animations and simulations that might be very useful or understanding that idea. So this gives you sort of a variety of ways in which you can use a particular approach to not only build your own assessments, which then flow directly into your gradebook, but allows you to really see not only whether or not they're doing the assessments, not doing the activity, but how those particular assessments allow you to understand their progress, if you will. It'll allow you to see how many tries, it'll allow you to see what they got wrong, and it'll allow you to really understand where your students are at. So all of that, I think, gives you a tremendous amount of diagnostic power, if you will, in terms of what you're interested in learning. So, one of the interesting questions is, how does this kind of visual exploration driven by technology, driven by a platform, really help students understand processes more effectively? So something that we've done, this is built on a variety of things that I've done in the past, is to actually use this as an opportunity, to then take a look at what does this do for students. Let me enlarge this a little bit, so it's a little bit bigger. Oops. Okay. So one thing we were very interested in is, how does this work for students in terms of simple retention of concepts? And what we saw using two of the visual synthesis maps is that indeed, if we look at the blue bar and we look at the maximal possible learning gain in terms of score, what we saw with these two sets of concepts using the gene synthesis, the gene expression rather, map, and also using the map that looks at speciation, is that in terms of the simple retention of concepts, using the text alone is the blue bar, but then using the visual synthesis map alone, what we saw was a moderate gain in terms of simple retention. And this was simple retention done immediately after either the completion of a reading exercise or the completion of a 30-minute exploration of the visual synthesis map. So in terms of simple retention of, do you understand a term, do you know a definition, we saw a moderate gain with groups of students. What was quite striking, however, is that when it came to actually synthesizing concepts, placing things into context, but in particular applying it in the context of interpreting, for example, data or another visual representation, what we saw here, once again, with text alone and still diagrams, what we see, that's the sort of learning outcome, the learning score. Once again, for the two topics, a significant gain. On the left is gene expression, on the right is speciation, and what you can see is there's a significant jump in terms of gain, almost a doubling of the gain of a higher order Bloom's taxonomy kind of application and synthesis of concepts. What we also found is that with using animations in general, there is a striking gain as well in the interest in learning more. Something we're very interested in of late is not only are learning outcomes, measurable learning outcomes of central importance, but you want to have a motivational gain for students as well. And what we're seeing, and we've unpacked this in greater detail, is that the ability to actually explore self-paced and in their own direction, if you will, under their own choice, is incredibly important for students. They become much more engaged in the material, and it actually drives them to want to learn more about what they're seeing and the concepts that they've addressed. So that's something that we feel is sort of an incredibly important gain. And in fact, I'm very interested in exploring further how agency actually allows students to really tie in more strongly and feel more strongly interested in the learning outcome. So sort of in a different study that I've done in terms of the use of visualization learning over time, comparing text plus the use of sort of visualization of animation material, what I've seen is that over time, here we see with text alone in blue, the drop-off in retention and application over a period of 2 years, those students that worked with visual materials and interactive materials, as you can see, the decline in retention is significantly mitigated over a period of 2 years. So we do think that there is a real place for using technology to really understand and to really drive greater motivation and a much more effective, even initial and long-term sort of retention of concepts. So this is just a little bit of some of the things that I've been interested in for a number of years and have been exploring. What I'd like to close with in terms of me blathering on, part of the presentation, is also thinking about how does technology allow us to understand in real time how students are doing. So what I will speak to is the importance of using learning management systems as a way of getting much more on the fly or incremental sort of insights into what your students are doing. So let me give you one rather simple example. For much of my career, I would assign, very interesting readings, hopefully, particular activities students would do. I would tell them time and time again how important it is for them to do this prior to coming into lecture so that they would have an opportunity to then engage with the material in a much more interactive and problem-based way in lecture. Lo and behold, what I would find time and time again is that I would stand up in front of them wondering if I could assume that they had actually done it. And what I discovered is that, of course, you quickly discover that, our students are often trained to get away with sort of doing things either at the last minute or not doing them at all and then winging it. And so what I quickly discovered is that I would waste a chunk of my time in class reviewing extremely foundational things that I then needed to build on. So not even using video, what I was able to do is to unbundle my readings and load them into the learning management system. One can do the same thing here, for example, using the ebook on the platform that we have. So that, for example, what you can see with, with something like Launchpad is that you can, in fact, take advantage of the ebook. You assign things actually through the actual ebook itself. And what this allows you to do with this kind of structure, and I've also done it with learning management systems like Canvas, is that it allows you to not only assign the reading digitally, but it allows you to intersperse assessments throughout, so that this gives you the ability to then not only assign pre-lecture work, but to actually know in a very specific and granular way who has done it. Right? Who has allowed you to actually focus? How does it allow you to know that your students have done the work? How does it allow you to then know what sections, for example, did they have the greatest trouble with the assignments? How is it, for example, in Canvas, I can tell that something that I expected would have taken them 10 minutes actually took them 30 minutes. So that when I stand up in the class, I can actually start from a position of knowledge in terms of what they've been actually able to do and how they've spent time on it. So that's yet another very simple way, not necessarily using visualizations, using video, where technology can give you intel, if you will, into whether or not your students are doing the work in a very granularized way, but what they've been struggling with, so that you can then focus your time with them on much more interactive activities that start really at the right place. So that's something that I think is really a very important kind of a technological sort of opportunity for us that we should take advantage of. So there have been a number of things which I've played around with, but what I really want to do is to give you folks an opportunity to actually ask questions and for us to have a discussion, because I know from the number of you out there that there is a lot of expertise out there, and I want to hear from you, and we should use this as an opportunity to not just hear from me, but actually as a group, really start to think about what other folks have done. So let me sort of open up things right now.
And since you are, thank you very much, Rob. This is Scott Guile again from Macmillan. You can submit questions via chat. We've got we've got two that have come in. You can continue to submit questions via chat. I will read them, and Rob will address them. The first one, the maps are lovely. I assume that the visualization maps. The maps are lovely, but are they too much for the average biology student? I guess the assumption being that you're working at Harvard with these.
Of course, of course. So one thing, that's something I've been very interested in as well. To what degree does this work with a range of students? So one thing I should say is that I've done work with the maps, using my own students here at Harvard, which are actually very diverse in terms of the range of backgrounds that they bring to bear. But I've actually been working with high school students from a wide range of high schools in the Boston area using maps as a way to engage them as well. One thing I found, and it was one of our inspirations for taking this approach, is that with student-aged people, they are so accustomed to Google Maps that for them, this is completely second nature. This notion of diving, of panning, of moving around is something they're very accustomed to, either from Google Maps or from doing it in fun gaming environments. So what I found from focus groups that we've run is that students are in fact far more comfortable with this than instructors are comfortable with it. I think it's a generational issue. But what I've also done as well is to think about it in terms of an on-ramp. So one way of doing it is to, in fact, and that's why I think it's important for instructors to show it in the classroom, is to open it up in the classroom, show them one or two little pathways through, and then leave it to them to go exploring on their own time. And I also encourage them to do this in groups. So I found that actually a very wide range of students, I've seen it used very effectively, not only in my class, but I've done focus groups with high school students, and we have a project going, for example, where we're doing a series of collaborative classes with students from a two-year college in the Boston area. And so a wide range of students, I think, take to it, frankly, more quickly than instructors do. Okay, Scott, was there anything?
Yes, the question about another one about the visual synthesis maps. Are the visual synthesis maps available to test out, explore online, or only through adoption of the textbook? You can take that, Rob, or I could, I suppose.
Well, Scott, in terms of accessibility via the textbook route for this specific book, for our specific book, why don't you take that?
Okay, yeah, they are available to explore. You do not have to adopt them to get access, and you can, we're going to follow up with all the attendees after the webinar week, and if you want access to the Launchpad that has these, we will be happy to send it to you. So no, that you don't have to adopt the maps to use them, to play with them, to explore them. Another question about the data. Where is the data from, and how was it compiled? I assume that the data of the presentation.
Right. So there are a series of ongoing studies that we've been doing. So I have a group called BioVisions, which is a collaborative sort of group that's focused on science visualization and how it can really drive both learning and engagement more effectively, and how it can hopefully enhance it. And so something we've been doing over a number of years now is doing not just focus groups, but also A/B comparisons of both how students learn from text alone, from text and still diagrams, and from text and sort of animations, and also interactive work as well. So the newest set of stuff, which I've just shared some of it, is focused on the interactive piece, right? So in other words, how does the ability to self-explore a particularly responsive visualization enhance one's learning. So we are in the middle of doing the studies now. So what I've shown you is not published yet. It is just sort of a series of first studies. So the N that we're looking at for those studies is on the order of, it ranges from 35 to over 70 students per sort of experimental group, if you will, per A-B comparison. But what we're trying to do is to expand this more significantly, do it with more modes of visualization, and really make sure that what we see, what seems like a significant learning gain, actually holds true. There's quite a bit of published literature showing, if you will, the benefit of, for example, still diagrams that are more synthetic, but there's very little at this point that compares moving image in terms of visualization. And so that's something that we're interested in doing. In terms of the format of the studies, how things were done, what we would do is have two randomly selected groups of students. I'll just share with you one study. In one study, students were exposed to 2 hours with a standard sort of textbook and still diagrams, and then the other sort of group had 1 hour with the textbook and with animations or with interactive media. And so that was the A-B comparison. That was one example.
Thank you, Rob. Another one from attendee, do you tape your lectures and provide the audio with the video to students for later views and reviews?
Yes, we do. So this has been an interesting point of contention. So for example, with my classes, all of the classes are videotaped. So they have both my voice as well as what's happening on slide. We do quite a bit in terms of demonstrations and also active learning as well. So there's quite a bit of panning around of the video. I'm fortunate enough to have a videographer working with me in my classes. There's been a lot of debate about whether or not this has a negative impact on students coming to class. What we're found is that because we have a lot of active learning that happens in the classroom, because we do a lot with, for example, Poll Everywhere, with clickers, if you will, students actually come to class. We usually have at least, let's see, our very lowest attendance rate is 85% to 86%. And this is a class of about 500 students. But what we're found, though, is that students do use the video for, if you will, review, right? So they use it as an opportunity for review. That was what students always claimed for years. We didn't really have evidence, but using the learning management system, since we know when every student accesses a video, how long they spend with it, and when they access it, what we have seen is a surprisingly sustained accessing of video sort of throughout the term, throughout the semester, so that they are really using it for review. So that's something that we think is very valuable. There is a colleague of mine at the college that, and this is another example of a use of technology, that has been playing around, and I played around with it as well, with a software suite called Bright Class. And what Bright Class allows you to do is to load up video, your lecture video, and students actually in real time can annotate the video and highlight a section of the video on a timestamp as to where they're confused and ask questions. That is all shareable across the class, and then the instructor can actually look at that and either respond to it online or can respond to it, for example, in person at the next class. So that's another way in which video of your class, of your lecture, if you will, if you lecture a lot, can become a record that then is a substrate for the discussion of what was confusing, where there was a misconception or a misunderstanding.
Thank you, Rob. In a bit of a plug here, since I do work for Macmillan, we're actually developing, we have in beta a video tool that allows for that same capability, that you can take students and annotate it. So that's something that we'll be rolling out in the spring. We have another question. Again, it's kind of for either me or you. I think I will take this one. These maps are amazing. Thank you for whoever wrote that. Is there a similar map of the brain? And if so, might we have it for our psychology courses? And right now, there is not a map of the brain. I can tell you which ones there are, and maybe Rob could talk about which ones are in development. Right now, we have them for angiosperms, diversity, ecology, gene expression, genetics, harnessing energy, and speciation. Rob, you would probably know more about ones in development.
Right. So, I mean, we are in the process of developing further maps. I think a map of the brain would be remarkable. Where that's not on the immediate next round, but that's certainly something we should think about. I think it's actually great hearing this as we're working on the second edition. But for example, a map that we're working on now that we're very excited about is in fact a first map that will serve as a framing structure for actually not just one map, but a series of maps. It's essentially a map of the major organ and physiology systems of the human body. So the very first one that we're going to focus on is the kidney. And this will give us an opportunity to go from, you know, the gross level of kidney as organ, and then to go down to, for example, at the membrane level, the transport of, for example, salts across a membrane, and how ion pumps are involved in that process. So for me, you know, when I've taught transport in the kidney, it's always been at the very, if you will, molecular micro level. Then you have this zoom out to the whole organ system, which I've always just done verbally and with still diagrams. Now we'll be able to put the kidney in the body as an actual functional organ, but then they'll be able to zoom into it. They'll be able to go from kidney physiology down to membrane transport. Right, and that I think is going to be hugely exciting for us. It also be an opportunity for us to tie together not just the macro and the micro, but to also connect it to critical concepts like osmosis. You know, and critical concepts like, you know, membrane permeability. But by tying it together, it allows students to see how understanding the principle of membrane permeability, or more accurately, membrane impermeability to ions, is so crucial to understanding how the loop of Henle works in the kidney. So we are in the thick of that map right now, of that visual synthesis. It'll be a new one in the book, but it will also be a new, highly interactive map online as well.
And I can say from the Macmillan standpoint, we are the leading publisher of psychology texts in the undergraduate market. Generally, when we roll out a technology like this and it's successful, which this has been, we expand it to our other markets. So it's very likely that we will be offering these for the psychology class, and if we were to offer them for psychology, the Chapter 2, Chapter 3 brain would probably be the very first one on the list. I can't promise you that that's in dev, but in all likelihood, psychology is looking at these, and if they were to do them, brain would probably be the first one that did.
So Scott, I think what we should do, and of course we're sort of talking book stuff in front of everyone, but I think this is interesting. I mean, let me know, because I think this is an opportunity. You see, one of the powers of this kind of technology is that it allows you to create something or to use something that bridges not just concepts across one book, but concepts across fields. So for example, what we created in terms of speciation is something that, you know, not just for freshman biology, for courses in evolution. It's a very important kind of a synthesis map that they'll be able to use, and it's something that I'm hoping students will be able to carry over, if you will, from one course to another.
And generally speaking, we try to develop our media for specific titles. However, with how intensive these are to develop, it is likely that we will repurpose some of these for other areas, such as evolution or another course, depending on the content. And yes, we are talking book stuff in front of folks, but someone did actually ask that, so I don't feel too terribly guilty. I don't believe there's any other questions. Let me check chat one more time. And If there is not, I will wrap it up. Thank you all so much for your time today on a Friday afternoon. We appreciate it. Rob, you as well. This has been great. Have a lovely, hopefully long weekend. And the recording will be available starting next week, and we'll follow up with each of you. Take care.
Well, thank you, everyone, and have a good weekend.